Hello everyone. In this chapter, we are going to address chromosomes, specifically the structure and the number of chromosomes in our cells. Now, as we go through, we are going to take a look at the different parts of a chromosomes. What are the noticeable parts of a chromosomes that you need to remember? We are going to describe how to obtain chromosomes so that we can analyze them, right? From which kind of cells in the body can we obtain chromosomes? How can we detect these chromosomes? And how can we tell the different parts of the chromosomes apart? We are going to dive into atypical chromosomes in terms of number of chromosomes, whether an individual has too many chromosomes or too little amount of chromosomes. Also, we are going to dive into the structure of chromosome in terms of, well, is there some loss of segment of a chromosome? Is there some gain of some segments of the chromosomes? What could be the consequences in an individual's health, right? And also, and finally, we are going to look into situations when an individual has received extra chromosomes from one of the parents, or is missing chromosomes from one of the parents. So in either case, we are going to dive into all these different aspects of the chromosomes. So now the first step we are going to make is to talk about cytogenetics. Cytogenetics, as the name indicates, cyto means cell, genetics, it's basically the discipline that studies the genetic information that is in the cell. So, cytogenetics, more specifically, is going to focus on the changes that happens on the chromosomes so that we're going to ask, well, how do this change relate to some particular conditions, including illnesses, right? Some specific traits, okay? So most of the time, what we notice in the body is that excess of genetic material is going to be overall milder in terms of effects on one's health compared to situations when there is loss of genetic information. So there are defects that happen as cells divide, and particularly how meiosis occurs. You know that humans reproduce sexually, so the formation of gametes is going to be a central key to the outcome in terms of health and in terms of consequences as to the structure of chromosome, because there are rearrangement of chromosomes, the shuffling of chromosomes during meiosis, right? We saw this during meiosis, right? So we're going to see that, you know, there are really a lot of possibilities for chromosomal abnormalities, right? And these chromosomal abnormalities, most of the time, are going to disrupt or completely stop the prenatal development. In fact, most of spontaneous miscarriages are caused by either missing genetic information or rearrangement of genetic information so that the development is not happening properly. Now, believe it or not, only about 0.65% of all newborn have chromosomal abnormalities that produce symptoms. Why is that possible? Well, this is possible because most of the defects are going to be eliminated and are not going to allow to continue so that an individual is born abnormal. Again, most of the spontaneous miscarriages occur because, well, you can have extra chromosomes or a zygote that is missing an important amount of chromosomes. In either case, this is going to actually cause the death of an embryo that is trying to develop. All right, so now let's take a look at the chromosomes and the structure of the chromosomes. Now, there are three major parts on the chromosomes that I want you to remember. One, it's the telomeres. The telomeres are going to be at the very end of the chromosome, and because our chromosomes are linear, the two ends are going to have some areas that we call telomeres. What are telomeres? I'm going to address that in a minute. So you have the telomeres that are structural part of the chromosomes. Two, it's going to be the centromeres. The centromeres are the areas where, after duplication of the genetic information during the S phase, during interphase of the cell cycle, the two sister chromatids are going to stay connected together, right? So it's going to be in this area right here. This is where we have the centromere. The centromere, it's actually a bunch of proteins that are going to bring the two sister chromatids together so that they don't part ways. Very important. And finally, but not least, the last segment that is important in the structure of the chromosome are the origins of replications. Okay? The origins of replications are a very important segment of the chromosome because this is where obviously replication is going to start, right? And the genetic information is going to be copied so that cells can divide. Now, chromosomes, we use the word chromosomes when chromatin is so condensed that we can see the genetic information during the cell cycle and under a microscope, right? Now, I mentioned chromatin. Chromatin, it's basically DNA mingled with proteins, right? And the particular proteins I'm talking about are histones, right? Now, chromatins occur in two kinds. We have heterochromatin, right, that look dark under a microscope, and euchromatin that looks light. Heterochromatin, it's that chromatin that is so dense that actually the genes that are in these areas of chromatins are not available for gene expression. While in euchromatin, the chromatin is loose enough, even though it's mingled with histones, it's loose enough to be compatible for gene expression, right? Let me close that parenthesis. Then, other aspects, when we look at chromosomes, and particularly after replication, we look at the centromere, we see that chromosomes are going to be organized in such a way that you're going to have long arms and short arms. So the short arms on the chromosomes are called P arms, while the long arms of a chromosome are called Q arms, right? So that when we address chromosomes and when we look at the defects, we are going to ask the following question. Where in the chromosome is the defect occurring? Or where in a chromosome is a gene located? Is it on the P arm, which means short arm? Is it on the Q arm, meaning long arm? Where is it located, right? So, knowing the structure of the chromosome, we are going to know that when we say P, we say short arm. When we say Q, we say long arm. With that being said, chromosome, it's basically a coiled structure of DNA mingled with proteins. But let's go a little further here. Actually, it's not only and exclusively DNA and protein. Guess what? There is a small amount of RNA that is also found as structural part of the chromosomes, right? That sounds a little weird? No, actually, it should not sound weird. Why? Because, well, the cell is always metabolically active, right? You always have some gene expression. You always have some RNA somewhere, right? So it doesn't come as a surprise. Or at least, do not be surprised that we have a small amount of RNA here. Now, the chromosomes are going to be distinguished based on their shape and based on their size. I want you to remember that. So their shape and size are going to tell them apart. And finally, let me come back to telomeres. The telomeres at the extremity of the chromosomes are going to be sequences of DNA. Sorry. Sequences of DNA that are repeated. All right? And these sequences of DNA are sequences of TTAGGG. So TTAGGG are repeated multiple times. And this sequence is actually shortening every cell division. I was saying that telomeres are found at the extremity of chromosomes. These telomeres are repeat sequences of DNA. Specifically, it's TTAGGG repeated several times. During the cell cycle, this telomeres, so TTAGGG, are going to shorten, right? So as the cell divide, every single cycle, the telomeres are going to shorten to the point that the cell can reach a point where the telomeres are so short that now the part that encode proteins is starting to be attacked. At that point, the chromosomes start losing their integrity. At that point, the cell cannot divide anymore. That cell is going to die off, right? So let me close that parenthesis. The centromeres are basically where the sister chromatids are going to be connected together, right? Where the mitotic spindle are going to come and attach, right? There is a particular structure that we call the kinetochores. I'm not going to bug you with that, right? The centromeres are made up of a bunch of proteins. These proteins are going to recognize a particular area, right, that contains 171 base pair of DNA sequence, and these proteins are going to attach to these areas. These proteins are actually called centromer protein A, right? This centromer proteins A are going to attach there so that the two sister chromatids can come together, right? And believe it or not, during the S phase of interphase, these proteins are not only going to facilitate the replication and bringing the two sister chromatids together, but in addition, the centromer protein A, CNPA, are going to be passed down to the offsprings. And this is very important because down the line, the chromosomes will have to be copied again, and the sister chromatid will have to be brought together again. Think, for instance, of when you have a cut or when you have a burn and you need to replace dead cells or you need to repair the cut. All right. The centromers occur in different places of the chromosomes depending on the chromosomes. There are some centromers that are found at the very end of the two sister chromatids. This type of centromers are telocentric, right? That's their position. Then we're going to have centromers that are not quite at the extremity of the chromosomes, but they are almost at the extremity of the chromosome. These ones are going to be called acrocentric. So they are closer to the center, but not quite at the center though. Not quite at the center. Then we have the most common, actually, are the sub-metacentric. Sub, underneath, meta, the middle, right? Sub, underneath, meta, middle, centric. So they are close to the center, but they are not quite at the center, right? And these are the ones where we are going to clearly see some short arms and some long arms, okay? And then we have the metacentric centromers. The metacentric centromers are the centromers that are located at the very center of the chromosomes, right? All right. The telomers, I told you what the telomers are. The telomers are at the very end of the chromosomes. Now, the subtelomere area, it's an area that comes right before the telomere, right? And this area is very similar to the telomeres. It is made up of about 8,000 to 300,000 base pair, right? They are going to be repeated sequences, right? So while here you have the telomere, in the subtelomere, we are going to see that, well, we also have some repeats, okay? So for instance, we have TAAGGG, right? And then we have GGG, TTA, GGG, TTA. So you see it's a repeat, right? And these repeats are going to shorten every cycle that the cell undergoes. These repeats are going to shorten until actually the shortening is so much that some areas of the chromosomes that carry genes that encode specific proteins start being attacked. And this is when the cell cannot divide anymore. All right. So we already talked about karyotypes, but karyotypes are type of charts that show actually the chromosomes. It allows us to say how many chromosomes an individual has, if there are too many chromosomes or too little amount of chromosomes, if the chromosomes are abnormal in terms of shape and in terms of size. For instance, if a chromosome is too long, if a chromosome is too short, right? We are going to be able to tell this. However, what we can never tell is if there is a mutation in a chromosome. The kind of mutation I'm talking about, it's, for instance, a substitution, right? So A is replaced by C, or G is replaced by T, right? You cannot see that with a karyotype. But with a karyotype, you can tell if an individual has an extra chromosome, right? Or that individual is missing chromosomes, or that individual has a major deletion, or a major duplication, right, of the genetic information. So anyway, all right. So when we look at the chromosomes on a karyotype, we have 22 pairs of autosomes, right? Pairs of autosomes. So the pairs of autosomes are these chromosomes that are not involved in the determination of sex characteristics, right? In addition to this 22 pairs of autosomes, you're going to have two sex chromosomes. One is X, the other one is Y, right? So that at the end of the day, for the human species, right, we have 24 chromosome types. We have 24 chromosome types. Out of the 24 chromosome types, we have 22 pairs of autosomes, one X chromosome, and one Y chromosome. All right, so the picture here shows you actually the arrangement of chromosomes based on their size and their shape. Now, they are organized by homologous chromosomes. So pair number 1, it's one chromosome you receive from mom and one that you receive from dad. Pair number 2, so one you receive from mom, one that you receive from dad, right? Now, they are homologous because they contain the exact same genetic information at every single location, normally, right? Okay, so with that said, here, for instance, we do see that chromosome number 21 here, rather than having 2, we're having 3. This means that in this karyotype, this individual here has an extra chromosome. That's what we call Down syndrome, right? Down. Oops, no, that's not correct. Down syndrome. There we go. Or trisomy 21. All right. So how can we detect chromosomes? There are different techniques that we can use to detect chromosomes. There are some techniques that are rather old, but that are still in use today. And there are some techniques that are very modern. Now, when we want to study the chromosomes, look at the number of chromosomes and the structure of chromosomes, well, you can use virtually any cell in your body. However, the cells that you cannot use are red blood cells. Why? Well, you cannot use red blood cells to actually study, for instance, a karyotype, or try to study any other perform any other genetic analysis, for the reason that red blood cells are the only cells in your body that do not have a nucleus when they are mature. Okay, let me repeat that. Red blood cells, or erythrocytes, are the only cells in the human body that when they mature, they do not have a nucleus. That's why these cells cannot reproduce, right? But let me close that parenthesis. All other cells, white blood cells, skin cells, liver cells, muscle cells, nerve cells, kidney cells, you can use them to actually analyze your genetic information. So I did explain why you cannot use red blood cells and why you can use other type of cells. Now, what are the techniques that are used for detecting chromosomes? We have amniocentesis and chorionic villus sampling, or CVS. I'm going to address that. And these are the relatively old approaches that are still in use today. And we have newer approaches, including the cell-free fetal DNA testing. Now, the older approaches are not ideal because they carry risks of miscarriage, right? They do carry risk of miscarriage. From that perspective, it's not the best way to go as we consider the technology that we have today, obviously, right? So that's one. Two, it's also that, well, the amniocentesis and chorionic villus sampling are not the best suited technology because they are very invasive, right? They are very invasive. So from that perspective, it's not the best technology. So amniocentesis is typically carried out when a lady is pregnant and when she's about 15 to 16 weeks pregnant. And at that point, using an ultrasound, we're going to figure out if the baby is healthy, is moving. Well, moving, not really. This is not really when you can tell, but you can at least hear the heart beating, right? You can hear the heart beating, but very importantly, you are actually sucking the fluid from the amnios, right? So some fetal cells are suspended in the amnios, and that fluid, when it is sucked, is going to be sent in a lab. They're going to do cell culture, and from these cells, they are going to extract DNA so that they can analyze chromosomes. And again, this happens between weeks 15 and week 16 of pregnancy. That's for amniocentesis. For chorionic villus sampling, this is going to happen much earlier. And this is going to be done at week 10 and 10 to 12 week of pregnancy. So this is going to require not to use a needle to go through the belly, but go through the vagina, right? Where you have some kind of catheter that comes in. And obviously, the fetus is much younger. And because of the area, which is the chorion, area right here, where the sampling is done, this causes a major disturbance. And as a matter of fact, the CVS technology has higher chances for miscarriage compared to amniocentesis, right? So that's for the chorionic villus sampling. Now, just like with amniocentesis, the cells that are obtained are going to be sent to a lab. They're going to do cell culture, and then they're going to do extraction of chromosomes and analyze chromosomes that way. All right. Now, the reason why a lot of people are doing this chromosome analysis, it's because, particularly in the case of a lady, as she ages, the chances of having abnormalities in the chromosomes increases. When we say abnormality in the chromosomes, we mean in the gametes, when the gametes form, right? So this graph actually shows you that the chance of having a child with trisomy 21, right, increases in an exponential manner after 35 years old. Okay, so notice that between the age of 20 and 30, right, chances of having a child with trisomy 21 is very low. However, when a lady reaches the ages of 25, we see that chances of having trisomy 20, a child with trisomy 21 increases. Now, notice that as we go to 40, it goes even higher. At 45, it goes even higher, right? So that speaks to really the high-risk pregnancy when a lady reaches 35. In the United States, at least, whenever a lady is 40 years old or above 35, the pregnancy, regardless of how the pregnancy is happening, it is actually addressed as a high-risk pregnancy, regardless of if everything is going fine with mom or not. So it is classified a high-risk pregnancy. That way, bunch of tests are being done, including the trisomy 21 test. All right. So after we extract chromosomes, we have to stain them, right? We have to stain them to locate the genes. Where are the genes located on the chromosomes now? For a long time, we didn't really know how to tell where the genes are located in a chromosome, but nowadays it is possible to actually isolate chromosomes and be able to tell specifically where genes are located on chromosomes, right? So because of that specificity, we can have some patterns in the bands that we see on the chromosomes because of all the improvement in the staining techniques that we have these days. Now, here is the thing. We have a technique that is so effective this day that is called fluorescence in situ hybridization. Fluorescence in situ hybridization, or FISH. This FISH, it's actually a technique by which you obtain a probe of DNA. That DNA probe is going to be labeled with a fluorescent dye. And you can use different kind of fluorescent dye. They can be green, they can be blue, they can be red, they can be different colors. That makes it really, really tremendously good, because you can follow multiple genes at the same time, right? And then after this probes have been associated with a fluorescent dye, These probes are going to allow to attach to complementary strands of DNA. Okay? And when they are attached to complementary strands of DNA, then we are going to introduce the result. The reading is going to happen on a computer with actually the use of algorithms that allows us to actually see the chromosomes as if we could actually see them with our naked eye. Obviously, we don't see them with our naked eye, but it appears on the computer as if we could see them with our naked eye, showing us really at different locations where the probe went and attached, and we see different bands on the chromosome. It's just amazing to see that, because now you can see a chromosome with bunch of, marks on. So here you have some cells with chromosomes that are stained, right? You can see some stain with using the fishing technique. All right. Now, let's take a look at the cell-free DNA testing. The cell-free DNA testing, it's a technology that has actually transformed how we actually track the fetus in terms of looking if they have the proper amount of genetic information. Now, how do we do that? Well, we just simply obtain some blood from mom. Okay? So we poke mom, get some blood, and that blood is going to be analyzed. Now, what we are going to look into, it's not the genetic information that is inside of the cells. Nope. We are going to take a look at the genetic information that is outside of the cell. Now, in the blood, we do have some DNA that is outside of the cell. And that DNA comes from cells that break apart, right? Say you have a white blood cell that breaks. That white blood cell that breaks is going to release its cytoplasm, and the nucleus is there. The nucleus is going to break down, and we are going to have some maternal DNA, right, in the blood. This maternal DNA, in general, is going to be made up of long fragments of DNA. However, we notice that in the blood of mom, when she's pregnant, she's also going to have some fetal cell-free DNA. But the difference is that these DNA molecules are going to be much, much, much shorter compared to the maternal cell-free DNA. Okay? So that's the difference. And now, we can tell if a baby or a fetus has extra chromosomes or is missing chromosomes. How can we tell? Because we are focusing on the proportion of the genetic information. So the proportion of the genetic information that is freely flowing in the blood. Okay? So the cell-free DNA, specifically the fetal cell-free DNA. Now, obviously, when we do this, we have to target very specifically some particular segments of DNA, right? Remember, we talked, for instance, about fishing, okay? So we can actually dye some fragments of DNA. All right. So, other aspects, when we look at the structure of chromosomes using karyotypes, we are going to be able to tell, which part of the chromosome is a long arm which one is a short arm, which arm carries a particular gene, right? Which chromosome number, right? So there are ways to actually write down on a report what we are specifically doing. So we're using some symbols and shorthand to describe what is going on with the chromosome and particularly the aberrations, right? So here is what it happens. After the shorthand, we are going to have the number, and that number is corresponding to the band that we see, right? And also the subband. So not only the band, but also the subband, right? We are going to address that in a minute. So when we look, for instance, at a particular individual, we run analysis of the chromosome, we are going to say that, okay, well, this individual has 46 chromosomes. Okay, so the karyotype says it has 46 chromosomes, 46 chromosomes. But wait a minute, it has 46, the individual has 46 chromosomes, but is that individual male or female? Well, if the individual is male, it's going to be 46XY. If the individual is female, it's going to be 46XX. That's how we know that the individual is a female, right? And that they have the normal amount of chromosomes. So there is no trisomy in that particular instance, right? So the bands and subbands are going to allow us to go to the gene, right? To be specific, for instance, is there a problem with the gene responsible for cystic fibrosis? Yes or no, right? Is there a problem with the gene responsible for sickle cell disease, right? Is that gene there? Yes or no, right? So we can tell that. All right. So how do we symbolize some abnormalities? Well, we are going to abbreviate 46XY for a normal male, 46XX for a normal female, 45X, 45 instead of 46, right? That means we are missing something, right? We say 45X, that individual is a female that is lacking an X, right? Well, we tend to say that female is a sex by default in human. Nevertheless, well, let me close that parenthesis. Not nevertheless, don't worry about this. Let me close that parenthesis. So 45X means that this individual does not have an X chromosome that they are missing, or they don't have a Y, right? That's a female with one X, nothing else. Then we have an individual that is 47XXY. So what do you say? 47, what does that mean? I'll let you figure out the answer. Okay, 47 means you have an extra chromosome. So 46 plus 1, that's 47, right? Now, the question is, which one is the extra chromosome, right? So we have XXY. Oh, this is a male that has an extra X chromosome, right? A male with an extra X chromosome. We can have 47XYY. This is a male with an extra Y. Then let's make things a little more complicated. We have 46XY del 7Q. Well, here we are saying this individual has 46 chromosomes, so that's good, right? He's a male. He's XY. Good, okay? He's male. But wait a minute. Del means that there is a deletion, okay? There is a deletion where on chromosomes number 7, but where specifically on chromosome number 7, Q, what is Q? Is Q the short arm or the long arm? The long arm, right? Okay, so this individual has a deletion on chromosome number 7. So he's missing some genetic information, right? This individual is 47XX plus 21 plus extra, right, chromosome in 21. So this is a female with an extra chromosome in number 21. So this individual has Down syndrome. Now, even a little more complicated here. So we have 46. Okay, good. Normal number of chromosomes. XY, this individual is male. T stands for translocation. Translocation means there is a shift, right? There is a shift. Translocation, right? Then we have 7, 9. There is something happening between chromosome number 7 and chromosome number 9, right? There is a translocation happening there. Let's be a little more specific. We have P and Q. Now let's focus on P first. We have P 21.1 and Q 34.4. What does this mean? This means that between chromosomes 7 and 9, there is translocation, first and foremost. 2, there are bands that are mingled, right? Not only that, but also legs, the short arm and the long arm. Now, let's be a little more specific here. So, we're saying at chromosome number 7, right, band 21.1, right? So, short arm of chromosome 7 at band 21.1 and long arm of chromosome number 9 at band 34.1. So we know specifically where the translocation is happening, right? Then finally, we do have 48. 48. What does that sound like? So normally we do have 46 chromosomes, but these individuals have 48 chromosomes. Obviously, we have some extra chromosomes here, but which chromosomes are extra, right? So now we have XX, YY. Oh, this individual, because he's Y, he has to be a male. Obviously, this male has not only an extra Y, but they also have an extra X, okay? A male with an extra X and an extra Y chromosome, right? So this is how you break down these things.